Hello everyone, welcome to our new section. Today we are going to discuss about the high throughput sequencing techniques such as Sanger sequencing technology and next generation sequencing. Myself, Sri Lakshmi, second MSc Biology from St. Mary's College, Thrissur. First, we can discuss about the RNA sequencing. It is a laboratory method used to learn the exact sequence or order of the building blocks that make up all the RNA molecules in a cell. RNA sequencing is done via direct method and indirect method. In the case of direct sequencing, it is done via the extraction of RNA, isolation of RNA, and finally the sequencing. But in the case of indirect sequencing, it is done via reverse transcription. Reverse transcription is a process which is opposite of transcription. It refers to the formation of complementary DNA, that is cDNA, using RNA as a template. In the case of transcription, we produce RNA using DNA as a template. In the same way, DNA sequencing is a method that is used to determine the precise order of nucleotides within a DNA molecule. There are several DNA sequencing methods such as Maxam and Gilbert method, Sanger's method, and next generation sequencing method. First, we can discuss about the Sanger's method. Sanger's method is also known as dideoxy or chain termination method. This method was developed by Frederick Sanger in 1977. It is a PCR-based method and also a modified DNA replication reaction. In this method or sequencing, the growing chains are terminated by dideoxynucleotide, that is DDNTP. In this Sanger sequencing method, we have primer, DNA template, DNA polymerase, normal nucleotides, that is DNTP, and dideoxynucleotide, that is DDNTPs. And mainly it involves 4 steps. First one is denaturation. Second one is primer attachment and extension of bases. Third one is termination. And fourth one is polyacrylamide gel electrophoresis. The Sanger sequencing have advantages as well as disadvantages. In the case of advantage, it is lowest error rate and it can target a primer. In the case of disadvantage, it is a long time consuming process and also have high cost per base. Next, we can discuss about the next generation sequencing, that is NGS method. It is a massively parallel sequencing technology that offers ultra high throughput and also speed. This technology is used to determine the order of nucleotides in the entire genome. There are mainly 3 steps in NGS method. First one is library preparation. In this, the libraries are created using random fragmentation of DNA followed by ligation with custom linkers. Second one is amplification. Here, the library is amplified using clonal amplification method or PCR. The third one is sequencing. DNA is sequenced using one of the several different approaches. The main concept behind NGS method is that the bases of small fragments of DNA are sequentially identified as signals emitted as each fragment is resynthesized from a DNA template strand. These are the steps of NGS method: DNA extraction, library preparation, sequencing, and analysis. Advantages of NGS method: low frequency variants can be detected with a higher sensitivity rate. Higher sample volumes have faster turnaround time. Comprehensive genomic coverage. Ability to detect the lower limit of samples, and hundreds and thousands of genes or gene regions can be sequenced simultaneously. Finally, the applications of NGS method: mutation detection, foreign DNA detection, non-invasive diagnosis aneuploidy, population characterization, cancer genetics, ancient DNA, expression analysis, transcription binding, and chromosomal interaction. Then, thank you.